Hello friends, warm welcome in this video, that is the glycolysis. Friends, as we all know that glucose is one of the most important compound in the plants, animals and microorganisms metabolism. This glucose possesses high potential energy and thus it is a good fuel to obtain energy. If we oxidize the glucose completely, it will convert into carbon dioxide and water with production of about 2840 kJ per mole free energy. Glucose can store in the cells in the form of starch or glycogen, and by doing so, it maintains the relative low cytosolic osmolarity. When in our cell energy demands increase, glucose can release from this storage form polymers and used to produce ATP. The ATP can be produced either in aerobic condition or also in anaerobic condition. Glucose is not only an excellent fuel, it also acts as a versatile precursor which provides number of metabolic intermediates for the biosynthetic reactions. You will get surprised if I say a bacterium like E. coli can obtain uric amino acid nucleotide, coenzyme, fatty acid or other metabolic intermediates needed to them from glucose for their growth. In animals, plants and microorganisms, glucose has four major fates. It may be used in the synthesis of complex polysaccharides like glycogen, starch, or it may oxidize to a 3-carbon compound like pyruvate via the glycolysis to ATP and metabolic intermediates, or it may oxidize via the pentose phosphate pathway, yield ribose 5-phosphate for nucleic acid synthesis and NADPH for reductive biosynthetic process. So, to obtain various intermediates and pyruvate, glucose has to split into smaller compounds. The process by which glucose converts into smaller 3-carbon compounds is called as glycolysis. In Greek, glykos means sweet or sugar and lysis means split, break. So the splitting of carbohydrates like glucose into simpler 3-carbon compounds like pyruvate is termed as glycolysis. The process of glycolysis occurs in the cytosol of a cell. Friends, glycolysis is an almost universal central pathway of glucose metabolism, the pathway with the largest flux of carbon in most cells. The glycolytic background of glucose is the sole source of metabolic energy in some mammalian tissues and cell types like erythrocytes, renal medulla, brain and sperm, some plant tissue such as potato tubers and some aquatic plants like watercress derive most of their energy from glycolysis, as well as many anaerobic organisms also are entirely depends on glycolysis. Friends, the history of glycolysis is also very interesting. The pathway of glycolysis as it known today took almost 90 to 100 years to fully elucidate. The combined results of many smaller experiments were required in order to understand the pathway as a whole. The first attempt to understand the pathway of glycolysis began in the 19th century with the wine industry. For some economic reasons, the French wine industry sought to investigate why wines sometimes turn distasteful instead of fermenting into alcohol. French scientist Louis Pasteur researched this issue during the 1815 and results of these experiments began the long road to elucidating the pathway of glycolysis. His experiments show that fermentation occurs by the action of living microorganism yeast, and the yeast glucose consumption decreases under aerobic conditions of fermentation in comparison to anaerobic condition. This is well known as Pasteur effect. Later on, insight into the component steps of glycolysis were provided by the non-cellular fermentation experiments of Edward Buchner during 1890. Buchner demonstrated that the conversion of glucose to ethanol was possible using a non-living extract of yeast, which is called as enzyme. This experiment not only revolutionized biochemistry but also allowed later scientists to analyze this pathway in a more controlled manner in the laboratory. In the series of experiments from 1905 to 1911, scientists Sir Arthur Harden and William John Young discovered more information of glycolysis. They discovered the regulatory effect of ATP on glucose consumption during alcohol fermentation. They also shed the light on role of one compound as a glycolysis intermediate, that is the fructose 1,6-bisphosphate. The elucidation of fructose 1,6-bisphosphate was accomplished by measuring CO2 levels when yeast juice was incubated with glucose. CO2 production increased rapidly and then slowed down. Harden and Young noted that this process would restart if an inorganic phosphate was added to the mixture. Harden and Young deduced that this process produced organic phosphate esters, and further experiments allowed them to extract fructose 1,6-bisphosphate. In 1906, Arthur Harden and William Young deduced the importance of enzyme cofactors, ATP, and other low molecular weight substances in fermentation. They tested their hypothesis that inhibitors of proteolytic enzymes would stabilize the glucose fermenting enzymes in yeast extract. They added blood serum known to contain inhibitors of proteolytic enzymes to yeast extract and observed the predicted stimulation of glucose metabolism. However, in a control experiment intended to show that boiling of the serum destroyed the stimulatory activity, they discovered that boiled serum was just as effective at stimulating glycolysis. Careful examinations and testing of the contents of the boiled serum revealed that inorganic phosphate was responsible for the stimulation. Harden and Young soon discovered that glucose added to their yeast extract was converted to hexose bisphosphate. They eventually identified the hexose bisphosphate as a fructose 1,6-bisphosphate. This was the beginning of the long series of investigations on the role of organic esters and anhydride of phosphates in biochemistry, which has led to our current understanding of the central role of phosphoryl group transfer in biology. Hans von Euler-Chaplin and Otto Warburg elucidate the whole pathway in essays, and Otto Meyerhoff, Gustav Embeden, Jakob Parnas elucidate the glycolytic pathway in the muscle cells of animals. As this pathway in muscles were discovered by Embeden, Meyerhoff, and Parnas, this pathway is also known as EMP pathway. Now we will see the summary of glycolytic pathway in short. The breakdown of 6-carbon glucose into 2 molecules of the 3-carbon pyruvate occurs in 10 steps. The first 5 steps are known as preparatory phase. In these reactions, glucose is first phosphorylated at the hydroxyl group on carbon number 6. The D-glucose 6-phosphate thus forms is converted to D-fructose 6-phosphate, which is again phosphorylated, this time at carbon number 1, to yield fructose 1,6-bisphosphate. For both phosphorylations, ATP is the phosphoryl group donor. Fructose 1,6-bisphosphate is split to yield 2 3-carbon molecules, that is dihydroxyacetone phosphate and glyceraldehyde-3 phosphate. This is the lysis step that gives the pathway its name. The dihydroxyacetone phosphate is isomerized to a second molecule of glyceraldehyde-3 phosphate, ending the first phase of the glycolysis. Note that 2 molecules of ATP are invested before the cleavage of glucose. There will be a good return on this investment. The energy will obtain in the payoff phase of glycolysis. Each molecule of glyceraldehyde-3 phosphate is oxidized and phosphorylated by inorganic phosphate and not by ATP to form 1,3-bisphosphoglycerate. The energy is then released as the 2 molecules of 1,3-bisphosphoglycerates are converted to 2 molecules of pyruvate. Much of this energy is conserved by coupled phosphorylation of 4 molecules of ADP. The net yield is 2 molecules of ATP per molecule of glucose used in anaerobic conditions and 8 molecules of ATP in aerobic condition. The energy is also conserved in payoff phase in the formation of 2 molecules of the electron carrier NADH per molecule of glucose. So friends, now we will learn each step in detail. Friends, the first step is phosphorylation of glucose. So why glucose undergoes phosphorylation? Because phosphoryl group seems to have 3 functions. That is, the plasma membrane generally lacks transporters for phosphorylated sugars. The phosphorylated glycolytic intermediates therefore cannot leave the cell. Thus, after the initial phosphorylation, no further energy is necessary to retain phosphorylated intermediates in the cell, despite the large difference in their intracellular and extracellular concentration. Second reason is phosphoryl groups are essential components in the enzymatic conversion of metabolic energy. The energy released in the breakage of phosphoanhydride bonds, such as those in the ATP, is partially conserved in the formation of phosphate esters such as glucose 6-phosphate. High energy phosphate compounds formed in glycolysis donate phosphoryl groups to ADP to form ATP. And the third reason is that binding energy resulting from the binding of phosphate groups to the active site of enzyme lowers the activation energy and increases the specificity of enzymatic reactions. The phosphate groups of ATP and the glycolytic intermediates form complexes with magnesium ions, and the substrate binding sites of many glycolytic enzymes are specific for these magnesium ions complexes. Most glycolytic enzymes require magnesium ions for their activity. So, in the first step of glycolysis, glucose is activated for subsequent reactions by its phosphorylation at carbon number 6 to yield glucose-6-phosphate with ATP as the phosphoryl donor. This reaction is irreversible under intracellular conditions. It is catalyzed by hexokinase. Hexokinases are the enzymes that catalyze the transfer of the terminal phosphoryl group from ATP to an acceptor nucleophile. And kinases are subclass of transferases. The acceptor in the case of hexokinase is hexose, normally D-glucose, although hexokinase also catalyzes the phosphorylation of other common hexoses such as fructose and mannose in some tissues. Hexokinase, like many other kinases, requires magnesium ions for their activity because the true substrate of the enzyme is not ATP but magnesium ATP complex. Magnesium ion sets the negative charge of the phosphoryl groups in ATP, making the terminal phosphorus atom an easier target for nucleophilic attack by an OH group of glucose. Hexokinase undergoes a profound change in shape and induced fit when it binds to the glucose. Two domains of the proteins move about 8 angstrom closer to each other when ATP binds. This movement brings bound ATP closer to a molecule of glucose also bound to the enzymes and blocks the access of water, which might otherwise enter the active site and hydrolyze the phosphoanidride bonds of ATP. Like the other 9 enzymes of glycolysis, hexokinase is also soluble cytosolic protein. Hexokinase is present in nearly all organisms. The human genome encodes 4 different types of hexokinases, that is, hexokinase first to hexokinase 4, all of which catalyze the same reactions. Friends, 2 or more enzymes that catalyze the same reaction but are encoded by different genes are called as isozymes. One of the isozymes present in hepatocytes, that is the liver cells, hexokinase 4, which is also called as glucokinase, differs from other forms of hexokinase in their kinetic and regulatory properties. So the second step is isomerization of glucose-6-phosphate or conversion of glucose-6-phosphate to fructose-6-phosphate. Enzyme phosphohexose isomerase, which is also called as phosphoglucose isomerase, catalyze the reversible isomerization of glucose-6-phosphate to fructose-6-phosphate. Glucose-6-phosphate is an aldol sugar, whereas the fructose-6-phosphate is a ketose sugar. The mechanism for this reaction involves an indiol intermediate. The reaction proceeds readily in either direction, as might be expected from the relatively small change in the standard free energy. This isomerization has a critical role in the overall chemistry of the glycolytic pathway, as the rearrangement of the carbonyl and hydroxyl groups at carbon number 1 and carbon number 2 is necessary to the next 2 steps. That is, the phosphorylation that occurs in the next reaction requires that the group at C1 first be converted from a carbonyl to an alcohol, and in the subsequent reaction, cleavage of the bond between carbon number 3 and carbon number 4 requires a carbonyl group at carbon number 2. Friends, the third step is phosphorylation of fructose-6-phosphate to fructose-1,6-bisphosphate. This reaction is catalyzed by phosphofructokinase first or PFK first. This enzyme catalyzes the transfer of phosphoryl group from ATP to fructose-6-phosphate to yield fructose-1,6-bisphosphate. Friends, many times students write the name of fructose-1,6-phosphate incorrectly. They write it as a fructose-1,6-diphosphate. Remember that compounds that contain 2 phosphate or phosphoryl groups attached at different positions in the molecule are named as bisphosphate or bisphospho compounds. For example, fructose-1,6-bisphosphate and 1,3-bisphosphoglycerate. On the contrary, compound with 2 phosphates linked together as a pyrophosphoryl group are named as diphosphate. For example, adenosine diphosphate, that is ADP. Similar rule apply for naming the triphosphate such as inositol 1,4,5 triphosphate and triphosphates such as adenosine triphosphate ATP. Friends, The enzyme that forms fructose 1,6-bisphosphate is called as phosphofructokinase 1. To distinguish it from second enzyme PFK second, that is phosphofructokinase second, that catalyzes the formation of fructose 2,6-bisphosphate from fructose 6-phosphate in a separate pathway. The reaction which is catalyzed by phosphofructokinase 1 is essentially irreversible under cellular conditions, and it is the first committed step in the glycolytic pathway. Glucose-6-phosphate and fructose 6-phosphate have other possible fates, but fructose 1,6-bisphosphate is targeted only for glycolysis. Some bacteria and protists and perhaps all plants have a phosphofructokinase that use pyrophosphate and not ATP as the phosphoryl group donor in the synthesis of fructose 1,6-bisphosphate. Phosphofructokinase first is subject to complex allosteric regulation. Its activity is increased whenever the cell's ATP supply is depleted or when the ATP breakdown products, that is ADP and AMP, accumulated. The enzyme is inhibited whenever the cell has ample amount of ATP and is well supplied by other fuels such as fatty acids. In some organisms, fructose 2,6-bisphosphate, don't confuse it with phosphofructokinase 1 reaction product fructose 1,6-bisphosphate, is potent allosteric activator of phosphofructokinase 1. Ribulose 5-phosphate is an intermediate in the pentose phosphate pathway. So the fourth step is cleavage of fructose 1,6-bisphosphate, that is the reverse aldol condensation of fructose 1,6-bisphosphate. The enzyme fructose 1,6-bisphosphate aldolase, which is often simply called as aldolase, catalyzes a reverse aldol condensation reaction. Fructose 1,6-bisphosphate is cleaved to yield two different triphosphate, that is glyceraldehyde-3 phosphate and aldose and dihydroxyacetone phosphate, a ketose. Friends, there are two main classes of aldolases. Class first aldolases found in animals and plants, whereas class second enzymes are found in fungus and bacteria. Class first enzymes form the chip base intermediates, whereas class second do not form the chip base intermediates. Instead, a zinc ion at the active site is coordinated with the carbonyl oxygen at carbon number 2. The zinc ion get polarized the carbonyl group and stabilize the enolate intermediate created in the carbon-carbon bond cleavage step. Although the aldolase reaction has a strongly positive standard free energy change in the direction of fructose 1,6-bisphosphate cleavage, at the lower concentration of reactions present in cells, the actual free energy change is small and the aldose reaction is readily reversible. The fifth and last step of preparatory phase is isomerization of glyceraldehyde-3 phosphate. Only one of the two triphosphates formed by aldolase glyceraldehyde-3 phosphate can be directly degraded in the subsequent steps of glycolysis. The other product, dihydroxyacetone phosphate, is rapidly and reversibly converted to glyceraldehyde-3 phosphates by the fifth enzyme of the glycolytic sequence, that is a triose phosphate isomerase. The reaction mechanism for this reaction is similar to the reaction promoted by phosphohexose isomerase. After the triose phosphate isomerase reaction, the carbon atoms derived from carbon 1, carbon 2 and carbon 3 of the starting glucose are chemically indistinguishable from carbon number 6, carbon number 5 and carbon number 4 respectively. The two halves of glucose have both yielded glyceraldehyde-3 phosphate. This reaction completes the preparatory phase of the glycolysis. The hexose molecule has been phosphorylated at a carbon number 1 and carbon number 6 and then cleaved to form two molecules of glyceraldehyde-3 phosphate. In this way, the preparatory phase reaction completes. In the preparatory phase of glycolysis, the hexose molecule has been phosphorylated at carbon number 1 and carbon number 6 and then cleaved to form two molecules of glyceraldehyde-3 phosphates. The second phase of glycolysis is payoff phase, which yields ATP and NADH. The payoff phase of glycolysis includes the energy conserving phosphorylation steps in which some of the chemical energy of the glucose molecule is conserved in the form of ATP and NADPH. Friends, remember that one molecule of glucose yields two molecules of glyceraldehyde-3 phosphate and both halves of the glucose molecules follow the same pathway in the second phase of glycolysis. The conversion of two molecules of glyceraldehyde-3 phosphate to two molecules of pyruvate is accompanied by the formation of 10 molecules of ATP from ADP. However, the net yield of ATP per molecule of glucose degradation is only of 8 because 2 molecules of ATP were invested in the preparatory phase of glycolysis to phosphorylate the two ends of the hexose molecule. So the very first step in the payoff phase is oxidation of glyceraldehyde-3 phosphate. Glyceraldehyde-3 phosphate get oxidized to 1,3-bisphosphoglycerate. This reaction is catalyzed by glyceraldehyde-3 phosphate dehydrogenase. This is the first of the two energy conserving reactions of glycolysis that eventually lead to the formation of ATP. The aldehyde group of glyceraldehyde-3 phosphate is oxidized not to a free carboxyl group but to a carboxylic acid anhydride with a phosphoric acid. This type of anhydride is called as acyl phosphate, which has high standard free energy of hydrolysis. Much of the free energy of oxidation of the aldehyde group of glyceraldehyde-3 phosphate is conserved by formation of acyl phosphate group at carbon number 1 of a 1,3-bisphosphoglycerate. Glyceraldehyde-3 phosphate is covalently bound to the dehydrogenase during the reaction. The aldehyde group of a glyceraldehyde-3 phosphate react with the SH group of essential cysteine residue in the active site in a reaction analogous to the formation of hemiacetal, in this case producing a thiohemiacetal. Reaction of the essential cysteine residue with a heavy metal such as mercury irreversibly inhibits the enzyme. The amount of NAD in a cell, which is equal to 10 raised to power minus 5 molar, is far smaller than the amount of glucose metabolized in a few minutes. Glycolysis would soon come to a halt if the NADH formed in this step of glycolysis were not continuously reoxidized and recycled. The second phase of payoff phase is a substrate level phosphorylation of 1,3-bisphosphoglycerate, that is phosphoryl transfer from 1,3-bisphosphoglycerate to ADP. The enzyme phosphoglycerate kinase transfers the high energy phosphoryl group from the carboxyl group of 1,3-bisphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. Note that phosphoglycerate kinase is named for the reverse reaction in which it transfers a phosphoryl group from ATP to 3-phosphoglycerate. Like all enzymes, it catalyzes the reaction in both directions. This enzyme acts in the direction suggested by its name during gluconeogenesis and during photosynthetic CO2 assimilation. In glycolysis, the reaction it catalyzes proceeds in the direction of ATP synthesis. Phase first and second of payoff phase of glycolysis together constitute an energy coupling process in which 1,3-bisphosphoglycerate is the common intermediate. It is formed in the first reaction and its acyl phosphate group is transferred to ADP in the second reaction. The energy released on oxidation of aldehyde to a carboxyl group is conserved by the coupled formation of ATP from ADP and inorganic phosphate. The formation of ATP by phosphoryl group transfer from a substrate such as 1,3-bisphosphoglycerate is referred to as substrate level phosphorylation to distinguish this mechanism from respiration linked phosphorylation. Substrate level phosphorylation involves soluble enzymes and chemical intermediates like 1,3-bisphosphoglycerate in this case. Respiration linked phosphorylation on the other hand involves membrane bound enzymes and transmembrane gradient of protons. The third step of payoff phase is conversion of 3-phosphoglycerate to 2-phosphoglycerate, that is the isomerization of 3-phosphoglycerate. The enzyme phosphoglycerate mutase catalyzes a reversible shift of the phosphoryl group between carbon number 2 and carbon number 3 of glycerate. Magnesium ion is essential for this reaction. This reaction occurs in two steps. A phosphoryl group initially attached to histidine residue of the mutase enzyme is transferred to the hydroxyl group at carbon number 2 of 3-phosphoglycerate, forming 2,3-bisphosphoglycerate. The phosphoryl group at C3 of 2,3-phosphoglycerate is then transferred to the same histidine residue, producing 2-phosphoglycerate and regenerating the phosphorylated enzyme. Phosphoglycerate mutase is initially phosphorylated by phosphoryl transfer from 2,3-bisphosphoglycerate, which is required in small quantities to initiate the catalytic cycle and is continuously regenerated by that cycle. The fourth step of payoff phase is dehydration of 2-phosphoglycerate to phosphoenol pyruvate. This is the second glycolytic reaction that generates compound with a high phosphoryl group transfer potential. Enolase promotes reversible removal of molecule of water from 2-phosphoglycerate to yield phosphoenol pyruvate. The mechanism of enolase reaction involves an enolic intermediate stabilized by magnesium ions. The reaction converts a compound with a relatively low phosphoryl group transfer potential to one with high phosphoryl group transfer potential. The fifth step of payoff phase and the final step of glycolysis is the transfer of phosphoryl group from phosphoenol pyruvate to ADP. In this step of glycolysis, the transfer of phosphoryl group from phosphoenol pyruvate to ADP is catalyzed by pyruvate kinase enzyme, which requires potassium ions and either magnesium or manganese ions. In the substrate level phosphorylation, the product pyruvate first appears in its enol form and then tautomerizes rapidly and non-enzymatically to its keto form, which is predominant at pH 7. The overall reaction has a large negative standard free energy change due to the large part to the spontaneous conversion of enol form of pyruvate to the keto form. About half of the energy released by phosphoenol pyruvate hydrolysis is conserved in the formation of the phosphoenolate bond of ATP, and the rest constitutes large driving force pushing the reaction towards ATP synthesis. So friends, in this way the aerobic pathway of glycolysis completes. So now we can construct a balance sheet for glycolysis to calculate the input of inorganic phosphate and ADP and output of ATP during the glycolysis. Friends, in the first step of glycolysis, glucose is converted into glucose-6-phosphate where one molecule of ATP is utilized. Similarly, in the third step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate by utilizing one molecule of ATP. In this way, the two molecules are utilized in the glycolysis in the preparatory phase. In step number 6, glyceraldehyde-3 phosphate is converted into 1,3-bisphosphoglycerate by net production of NADH. NADH, after entering into electron transport chain, yields approximately 3 molecules of ATP. Here, in the glycolysis of glucose, 2 molecules of glyceraldehyde-3 phosphates are converted into 2 molecules of 1,3-bisphosphoglycerate, and thus 2 molecules of NADH are generated, and therefore overall 6 ATP are formed in this step. In the glycolysis, 1,3-bisphosphoglycerate get converts into 3-phosphoglycerate. During this step, 1 ATP is formed, but as we have mentioned in step number 6, here also 2 1,3-bisphosphoglycerates are converts into 3-phosphoglycerates and thus generates 2 ATP. Friends, in the last step of glycolysis, that is the conversion of phosphoenol pyruvate to pyruvate, 1 ADP is converted into ATP. As we know, 2 molecules of glyceraldehyde-3 phosphates are entering in the glycolysis from the 6th step, and thus we will get 2 molecules of ATP during this process. So the overall ATP generated during the glycolysis are 10, but as we are utilizing 2 ATP during the preparatory phase, so the net gain is 10 minus 2, that is the 8. Friends, if you like this video, please subscribe to our channel, press the bell icon, and don't forget to share this video among your friends. Thank you.